Aloha, scholars. Welcome to APHY 102, and we are going to kick things off by talking about the action potential and the path that leads to muscle contraction. So, the very first thing that we need to talk about are two very important fibers, and these guys are called actin and myosin. Myosin is a really thick fiber, actin is a really thin one, and you have a whole bunch of these fibers in your muscle cells. And basically, what ends up happening is that one tugs on the other, and that's how you're able to move. Now, we're going to spend the rest of this video talking about how that process actually takes place because it turns out to be really important. The reason it turns out to be important isn't just because we want you to have to understand a lot of really complicated chemistry, although there is a lot of complicated chemistry in it, so yay. But as you can imagine, because of the complicated nature of it, that's where things like drugs are going to come into play. The legal kind, just to clarify. So these first couple slides here just show you a review table of the different structures of muscle. You can review these on your own. I kept this in here because I wanted to introduce actin and myosin and actually show you what they look like. Actin is a really thin type of filament. Myosin are really thick ones. And basically when these filaments slide against one another, that's what causes your muscles to move, leading to muscle contraction. And that's true in your skeletal muscles, in your smooth muscles, which are in your visceral organs like your liver and your intestines and your tummy, all those, as well as your heart. Although the process in each of those muscles is a little bit different. So what ends up happening is that the thin filaments slide over the thick ones so that actin and myosin overlap. Now, it is noteworthy that neither actin nor myosin change length during muscle contraction. So, myosin heads on the thick filament attach to myosin binding sites on actin. So, I think here you can see these myosin heads here. These myosin heads are going to bind to actin, and the big thick myosin filaments are going to tug on the actin in a little bit of a tug of war, leading to muscle contraction. Now, you're going to end up having lots of these things called cross bridges, and these are going to form and break a whole bunch as muscle contraction takes place. The other thing is that this is going to happen simultaneously throughout all the different fibers in the cell. Basically, all of these different fibers are going to contract at the same time, which is a good thing. This is not like a domino effect thing where one does another, does another, does another. It all tugs at the same time, because if it didn't, movement would be really inconvenient and horrible. Now, in order for muscle contraction to happen, though, there are a lot of things that need to happen along the way. The first is a stimulus. You do not want your muscles to contract on their own without a stimulus. For the most part, there's some exceptions, one of which is your heart. Your heart pretty much beats on its own without any additional stimulus. Another example of this would be in your smooth muscle, in your gastrointestinal tract. You actually have pacemaker cells in there that will pretty much self-stimulate. So there are exceptions where you do not need a stimulation for muscle contraction to happen, but another name for muscle contractions without a stimulation would be like a spasm, and that is not a great time. So your muscles basically are not going to contract without some sort of stimulation. So there are two things that must happen in order for a muscle to move. And you probably have not really seen a horse race. Maybe you have. Oh, yeah, we're in southern Indiana. You've definitely seen a horse race. It's like Derby's huge down there. So basically two things happen. The jockey, or the dude on the horse, cracks the whip, which is basically the stimulus, and then the horse moves. However, you need to have two things. You need to have a stimulus, which is the crack of the whip, And then you also need to have the energy for contraction to take place. So if you just, hit the whip against a dead horse, there's a stimulus, but there's not going to be any movement there because the horse doesn't have the energy to move. And that energy comes in the form of ATP. So, in order for a muscle cell to contract, it must be stimulated by a nerve fiber. So, welcome back, action potentials. So, the electrical potential or the electric current or the action potential is generated, then it gets propagated throughout the muscle cell, and calcium turns out to be a really important ion. So just to review the action potential, we talked a lot about it in 101, but I never made a video about it. So I thought I would take a stab at teaching you, and since it's been a minute since you've watched those videos, I thought now would be a great time to get things going. So the action potential starts at the axon, which is the long part of many neurons. Oftentimes it's myelinated, and that myelin sheath is going to insulate the axon and help the electricity get where it needs to go. But the axon divides as it enters muscle, and it forms what's called a neuromuscular junction, which sounds really complicated. It's really not. It's literally just the point where the neuron or the nerve fiber interacts with a muscle. That's it. That's all it is. And at this neuromuscular junction, you have what's called a synaptic cleft. And inside the synaptic cleft are chemicals called neurotransmitters that are basically just hanging out until they're told to do something. So the action potential arrives at the neuromuscular junction, and inside the axon terminal is these synaptic vesicles. So think of synaptic vesicles as being like these little water balloons that aren't filled with water. In this case, they're filled with a neurotransmitter. And the neurotransmitter that we often see in muscle fibers is acetylcholine, or ACH. So when the action potential arrives, or the nerve impulse, it releases acetylcholine into the synaptic cleft, which diffuses across and attaches to receptors on the muscle fiber. So when acetylcholine binds to the receptors, what ends up happening is that opens up these things called ligand-gated channels. Ligand-gated channels are just like a chemical. So acetylcholine is the ligand. So think of the ligand as being the key that unlocks the lock. And the lock allows for sodium to flood the cell, which changes the electrical charge of the cell from negative to positive, and the action potential is able to travel. So all of this that we're talking about so far is the action potential is basically the whip. The action potential is the stimulus that says, hey, move. And you don't want those stimuli to stick around for very long. It's not great to have your muscles constantly twitching. Another word for that is called a spasm. I don't know if you've ever had a spasm before. Think like a Charlie horse. Charlie horse is a spasm, extremely painful. And, because these fibers are very innervated and they communicate very heavily with your brain. So to keep these action potentials not lasting forever, you have an enzyme called acetylcholinesterase, which is going to degrade acetylcholine, which will limit the amount of action potentials at one site. So this slide is just a review. The action potential arrives, acetylcholine is released. When that happens, acetylcholine binds to the receptor, which allows sodium to flood the cell, causing depolarization, meaning the cell membrane is no longer polarized or no longer different than the outside of the cell. Remember, at nervous and muscular tissue, the cells are often polarized, meaning they have a negative charge at the plasma membrane. So depolarization is when that negative charge goes away, because a whole bunch of positively charged sodium are flooding the cell. That local depolarization ignites the action potential. So the next phase is called excitation contraction coupling. So the action potential travels all throughout the entire plasma membrane, or sarcolemma, that's what that is, of the muscle fiber. It travels along these structures called T tubules, and it releases this thing called calcium, these ions. And what ends up happening is that calcium binds to troponin. Now, troponin turns out to be really, 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 really important. Um, I'll post a link for a reading that I would like for you to do, but troponin is basically it is very common in the heart, and we see it a lot of times heavily elevated in someone who's going through a myocardial infarction or a heart attack. Troponin is usually that biomarker there that we see in when your heart is essentially spasming or beating out of control. So when calcium binds to troponin, myosin binding sites are exposed. So again, troponin is kind of like a lock. When calcium is around, calcium binds to troponin. Troponin gets kicked off of myosin binding sites so that actin can be exposed, and myosin will bind to actin, leading to muscle contraction. So pretty complicated slide, but this basically shows you everything that we've talked about. The action potential arrives. Calcium allows for acetylcholine to be released. Acetylcholine diffuses out of the synaptic vesicle, binds to a receptor on the cell here. When acetylcholine binds, it opens up sodium channels, which is able to depolarize the cell. So that's pretty much it as far as the action potential goes. The action potential gets transmitted throughout the muscle fibers, and that releases calcium. As calcium binds to troponin, troponin gets kicked off so that myosin is able to bind to actin, is pretty much the gist of it. And again, the thing I want to point out here, calcium binds to troponin. Troponin kicks off tropomyosin. Tropomyosin is actually the thing that's blocking the myosin binding sites. This is kind of a minor point, but if you read in depth about it, you'll find that. So I want to clarify that real quick. But all of this that we're talking about is just the whip. So remember, I talked about how muscle contraction is kind of like a jockey on a horse. All of this is the jockey cracking the whip, telling the horse to move. The next video, I'll talk about what happens that actually allows the horse to move from an energy perspective, because right now this is just the stimulus. All right, so take care and stay in tune for the next video.